Okay. Now, on the program, there's actually no end time. Now, my flight's at 8 o'clock, so I have to be out of here by 8 o'clock, okay?
Just judge it by how people
Okay, all right. So I'm Kevin Smyth, and this is Justin Cullen. We come from Ireland, just across the sea, and I'm a chemical engineer. Justin's a process architect. What we're going to talk about today are ADCs. So be honest now, do people know what an ADC is? Can you raise your hand if you know what an ADC is? I got 2.5, somebody's not quite sure. Okay, 2, 3, 4, okay. Now okay, that's it, 5, okay. So ADCs are one of the biggest growth areas in the pharmaceutical industry at the moment. So what I'm going to, they're called antibody drug conjugates, and they're a subset of drug conjugates, which are a major growth area as well. It's just the antibody drug conjugates. Big pharma is spending billions and billions on it at the moment. Pfizer, late last year, spent $42 billion buying Seagen. You know what I mean? AstraZeneca announced this week they're spending $1.5 billion on an ADC facility in Singapore. There's more detail out there. It's growing much faster than ATMPs or cell and gene therapy. Because I work for a design house, IPS, American company. Before that, I worked for Jacobs for 30 years, and we know what's happening. We know what's happening in the market, okay, before you find out on LinkedIn and Fierce Pharma and stuff like that. We know cell and gene is plummeting, the investment's plummeting. We know ADCs is going through the roof. But why are we here at a Cleanroom conference talking about ADCs? Because everything we talked about over the past 2 days with excellent speakers applies to ADCs because they get injected into patients, yeah? But there are different phases. And the thing that I find interesting about ADCs now is because they're extremely potent. We're talking about containment down to 10 nanograms, and in some cases 1 nanogram per meter cubed. They're the OALs, and people can't get their head around what that means. It's tiny, you know what I mean? It's nanograms. And I could put up a slide to show exactly what that is. It's probably, you know, a grain of salt in the whole of Birmingham or something like that. That's how tiny it is. You cannot release that into the environment. But also, so when we're talking about ADCs and very high potent drugs, we need to protect the operator and the environment from this highly toxic material, but we also need to protect the patient from a GMP perspective because we need to make sure there's no bugs in it, you know what I mean? So that's what we're trying to do. So in my opinion, it does apply to clean rooms because you make them in clean rooms, but there's something extra in it. I think the previous speaker was talking about you know, containment and trying to keep everything out of the clean room, only put stuff in the clean room that you need to put in it, and you'll have most of the vessels outside, and then somebody raised a concern about leaks, yeah? The principle of high containment is you only have stuff in that room if it actually needs to be in it, and anything else you keep out, because once it goes into that room, you have to figure out how do I get it out because it's contaminated with this highly toxic stuff. So you really, to get your head around it, you need to think nuclear. That's what you need to be thinking. People are aware of the hazards with nuclear, with highly potent stuff. It's the same kind of mindset. And the reason I'm saying that is because I've worked on many ADC projects. I'm working on 4 at the moment. And when you're talking to people, they just can't get their head around, look, we need to contain this stuff. So when we're designing our systems, or how we deal with our waste, how we deal with our water, where the vents go, where are we sure and guaranteed that potent material has not gone? How do we contain it all? So that's just an introduction, okay? So this is a picture of me, okay? So I'm trying to relate that to clean rooms. I couldn't really, okay? But this is a sport we have in Ireland, which is hurling. So these things here are big sticks, all right? And you have, like a cricket ball, a leather ball, right, that you hit with that stick, and the aim is to get it into a goal or over a bar and not to contact your body, right? If it hits your body, it's really sore, right? And so when you go out and play that, you kind of do a risk assessment beforehand. Where should you be on the pitch, yes? If you're in goal, you're going to get hurt. If you're up the far end of the pitch, far away from the slitter is what we call it, yeah? The chances are, if you're me, nobody passes you the ball, so you leave the pitch with no bruises at all. And that's about height containment. It's all about risk assessment, yes? And you have to risk assess, but you have to keep the emotion out of the risk assessment. And that's what myself and Justin are going to talk about. Where do we really need to focus in the design? So I talked about, we work for the American company IPS. This is not a big sell. It's just when I was talking to people over the past couple of days, nobody heard of IPS, right? We're an American company. We do have an office in Birmingham, just across the road, actually. We have an office in Manchester. We have an office in Basel. But they are American, and so we have lots of offices in America, but we're growing in Europe because apparently there's so much building going on in Europe, so much investment. We've opened an office in Denmark, no surprise, because of all the work that's happening in Denmark with a certain company. And so that's the spiel about IPS, what we do. So what is an antibody drug conjugate? Basically what it is, I think there's a pointer on this. Yeah. It's a monoclonal antibody there. Now the payload is this little thing here, this little red thing here, or there. And what the purpose of and then they're joined together by what's called a linker, yeah? So the monoclonal antibody, the purpose of that is to get, this is the drug here that does the business, right? The purpose of that is to get it to the cancer cell, right? So normally with chemotherapy and stuff like that, I'm not a doctor by the way, but when you give somebody chemotherapy, therapy goes through the bloodstream and kind of destroys a lot around it, okay? This is kind of the magic bullet. So the purpose of the MABs is to get it to the actual cancer cell, yeah, that it needs to treat. Once it gets to the cancer cell, the plan is that it's internalized within the cell, right? And then it's detached from the MABs, and this goes to work. Now, they are quite complicated because this linker sometimes detaches, sometimes doesn't, and it's all by design what happens. If that linker detaches, sometimes this potent, highly potent thing can leave the cell and go on to neighboring cells, and that's what you don't want to happen a lot of the time because it can damage healthy cells. But there's 15 of these actually being licensed at the moment globally, but they're not licensed in every country. So in the US, I think there's 13 licensed for use. I think in Europe there may be 15 licensed, something like that. But it's only been growing recently over the past 15 years. So we go to the next one. This is more just pretty pictures, okay? So there's your MABs there, there's your cytotoxic agent, and there's your linker. And this is just how it's internalized. So what you have is there's antigens like this on the surface of cells. So the idea is, hopefully, with a cell, a cancerous cell, a tumor like that, what they have is you have certain receptors that are more prolific on these cells. So the ADC, or the MABs, targets these cells and doesn't target the healthy cells. That's how it works. So with certain types of cancers, there's a lot of certain antigens on the surface of those cells, and they're targeted. And the ADC, as it's traveling through the body, just ignores all the healthy cells. It's simple as that. And that's just a pretty picture of what happens when it goes in, and there's different modes of action when it goes into the cells. So a historical perspective, this fella here, Paul, I can't pronounce it, Ulrich, yeah, he was born in Poland, but he lived in Germany for most of his life. And in around 1900, he came up with this phrase, okay? If a compound could be made that selectively targeted a disease-causing organism, such as a cancer cell, yeah, then a toxin for that organism could be delivered along with the agent of selectivity. So that actually describes an ADC. So he was thinking of that concept in 1900. So this guy was brilliant, okay? He won the Nobel Prize for medicine. He discovered a cure for syphilis before penicillin was brought in, okay? And he came up with this word, magic bullet. So that's any time you have somebody talking about ADCs, they always mention this person here. Now, I don't expect you to see this, but when I was looking at ADCs and I've been developing them, I say, well, what's the biggest cause of death in the EU? It's diseases, circulatory diseases, 33% in Europe, okay? But cancers, it's falling behind at 23%. And all ADCs at the moment, they're all dealing with oncology, okay? These cancer diseases and tumors, okay? So this is just a summary of what I talked about already. There's 13 approved in the US, 15 in total, but there's loads and loads of development being done. Over 400 are in different stages of development, all stages, and many of them in clinical trials at the moment, okay? And the focus area mainly is oncology, but there is a bit in rare diseases and hematology as well. So these are the ones here, okay? And what I did is I just, at the end of it, you see the different what they're for: leukemia, Hodgkin's disease. Now, Mylatag, for example, that was the first one that was approved in 2000, and then it was withdrawn in 2010, okay? And this is happening a lot with these ADCs at the moment. They're being withdrawn for various reasons because they are quite dangerous, yes? So the benefits versus the negative effects on patients can be quite severe. So Mylatag was then, I think, let's see what year it went. It was reapproved, right, in 2017, but for a lower dose of the drug and for a subset of leukemia patients. Now, there's some big blockbusters in here. I have a slide. These are the main companies out there at the moment who are investing billions, and they're acquiring companies. That's how they operate pharmaceuticals. They just acquire companies, and there's a lot of contract manufacturers out there as well, okay? Now, so Lonza, Millipore Sigma, that's the German work over in the States, Permel, the Indian company, SK Biotech, Korean company, Wuxi, Chinese company, Lotte, I can't remember where they're from actually, and Samsung Biologics. They're the big players in contract manufacturers, and why people go to contract manufacturers for these type of drugs is because they're so high potent, yes? And these companies have the skill sets to deal with these high potent. Now, I'm going to show you a few designs of typical facilities. I can't give you too much information, unfortunately, but what I'm trying to say in these slides is the ADCs are new, the design of the facilities are new. We are finding our way. And a lot in the pharmaceutical industry is subjective, emotional, and people's opinions, and it's not based on science. It's not like drug substance. I was involved in Pfizer Grange Castle. It was Wyeth at the time in 2000. We knew what we were doing, okay, because the company had built lots of big facilities like that. All these ADC facilities are totally different. The unit operations are more or less the same, but they are multi-product. They're all very, very small scale. You're talking about 60-liter reactors and stuff like that. You have to have isolation technology, but there's different types of isolation technology. You have a bulk fill operation. Some people want to do it under grade A conditions, which is absolutely crazy, you know what I mean? But you can't argue, okay, because it's in its infancy and you have different parties involved. But from a chemical engineer's point of view, what I try to focus on is containment and making sure we don't kill people outside, yeah? The GMP aspects of the drug, they're basics. We all know about that. But try not to kill people outside. Try to keep that highly potent material inside. So you won't be able to read this, but these were the sales, okay, in 2021 and 2022. And there's a few blockbusters that, there's a few blockbusters there which are in the red, and that was the growth in the sale of those, okay? So it went from 426 million to 1.2 billion for that and HER2. I think that's for breast cancer, yeah? So there's prostate cancer there as well. So, and these companies, they're building more and more facilities so they can make more and more of these products. So GMP and regulatory compliance. The next thing, because the drug consists of an API, the highly potent, it consists of a MABs. So the API, which is the highly potent plus the drug linker, that's synthesized in a conventional API synthesis plant. So that doesn't follow, Annex 1 or anything like that. It doesn't follow Annex 2. It follows less rigorous GMP requirements. Then you have your small molecule, not small molecule, your drug conjugate. That follows typically Annex 2 plus a bit of Annex 1, yeah, for a biologic, yeah, when you're doing the conjugation. When you do, sorry, I beg your pardon. Up here we have, that's your MABs, that's your API synthesis, and that's your conjugation. So your conjugation, which is bringing your MABs together with your API, that follows Annex 2 and a bit of Annex 1. And then after this, you need to fill it into vials, and that follows Annex 1. So, but typically, these different unit operations are carried out on different sites by different companies all over the world. Now, AstraZeneca, they announced this week, they're going to have to build a facility that's going to do all of this in the one campus. Yeah, all of it, and zero carbon as well, by the way, okay? I don't know how they're going to manage that, but that's their plan, okay? So the GMP requirements are different, you know, for this facility than this facility, than the final, the secondary packaging and the primary packaging. So you need to know, when you're designing these facilities, you need to be aware of everything. Because typically, I spent a lot of time designing fill-finish facilities, hadn't a clue about API synthesis. You know, it's just a dirty facility. But now you have to know everything. You can't over-design these facilities. So I have a picture here of, this was kind of covered before. You know what I mean? We need to get rid of the human. And for conjugation facilities, we need to do it for two reasons. One, to stop the human polluting the place. But two, we need to try not to kill the human as well in the place. So ideally, we need to use automation, and these are cobots, yeah? We should be doing them, okay? You know, there's Starbly, you know, the automation robotic company. They're all over the place. All filling machines are filled with their robots. So that's where we need to get to. We need to keep the human out of the clean rooms completely, yeah? And that's the way it will end up, because people don't want to work in them anyway, you know what I mean? So from a containment point of view, everyone's seen this chart, okay? You know, ideally what we want to do is eliminate the risk. We can't really do that because we need the risk, we need the highly potent stuff. But maybe we can change the form. Instead of having it in the powder form, which is the big challenging thing, have it maybe in some kind of crystal form like that, so there's less risk, okay? Can we substitute it? Can we use less hazardous solvents and stuff like that as part of the process? So a lot of these things we can't, as an engineering design house, we don't really have an opinion on. We can't change what they've done in R&D and stuff like that. We really come in during engineering controls and isolating people from the hazard. But we still maybe have an opinion. When you're looking at the process and the unit operations, for example, I was doing one project and the operator, the client had lots of different unit operations. They had synthesis followed by, I think it was what do they have? Synthesis followed by chromatography, followed by one form of drying, followed by another form of drying, and everything was a manual operation. And they said, well, we're going to dry it in this bit of equipment, then we're going to put it in a bucket and carry it and put it in this other bit of equipment to do more drying. And I said, why don't you use the one item of equipment to do both operations, so it kind of reduces the risk? And it's common sense. So you can't, the older you get, the braver you get. Because Keith was saying, look, there's no harm, always ask. You know, what can people say? No, that's all. Okay, so engineering controls is really where engineering design houses come in, and administrative and PPE. PPE is always the last resort. I think in Switzerland, some of the facilities, you know, in the world, you still see people wearing breathing air. In Switzerland, that's not allowed. You have to design it out. People should not have to wear breathing air. If people have to wear breathing air, you have failed in your design completely. And this is, if you ever are designing a plant, people keep going on about OEB levels, OEB 1, 2, 3, 4, 5, 6. There's about 50 different bandings. Yeah, so OEB 5 means absolutely nothing. You need to know what the OEL is. But anyway, any stuff got to do with ADCs, it's the top level anyway. It's extremely potent. That's it. Extremely potent. Okay. So look, there's legislation all over the world. You know what I mean? But the onus is always on the employer to protect the employee. But the onus is also on the employee to behave. You know what I mean? And especially with high potent. One of the sites I worked on, when you're trying to, if there's a spill or an incident with these materials, you have to react appropriately. And you'll see on these sites decontamination kits and special buckets all over the place. And some operators think, oh, we just have to pour this on the spill and it deactivates it and everything is safe. Well, that's okay for product A, okay? If you have product B, it may not work on it. So, you know, you have to question everything, because I was on the site and I was told that by somebody, oh, we use this material and we use it for any spill. So always question what's going on out there with high potency, because people just don't understand. So risk, I was talking about risk back on the hurling and stuff like that, so it's similar here. Risk is exposure by the hazard. The hazard is the OEL, there's nothing you can do about that. But the risk, okay, can be, you know, risk is exposure by hazard, yeah? The risk of exposure can be reduced, for example, if it's in liquid form. When it's in powder form, it's a big risk, you need to do it in an isolator. Once it's in solution, the OEL typically can go down. A lot of companies don't like that. They say, no, the band it's in remains band 6. That's crazy, right? But it's again trying to convince people that actually you can take, you know, you can base it on sound risk assessments, that you don't need to over-design these facilities as well. So what you'll see here, that's just an extraction booth, yeah? And that's where all liquid operations can take place in this facility. This here is a glove box, that's where powder is present, okay? Some companies wouldn't allow you to do that because they're saying, just because it's liquid form, we're still treating it as an OEB 6 operation, okay? I just like this picture, okay? So, but what I was trying to get here is liquid waste. That you get liquid waste from the process, you get liquid waste from CIP, from emergency showers there, okay, and from fire water. And one of the things that a lot of companies don't think about is fire water. So if there's a fire in one of these places, will the fire water be contaminated, and where does the fire water go? Now, that fire water needs to be contained and it needs to be tested prior to being released to the environment. That's not what happens, by the way, okay? So you always have to question everything. Also, typically, you'll collect this waste in special tanks, and those tanks will be in bonded areas, typically located outside. So you get rainwater in that bund. So what you do with that rainwater, do you treat that as potent waste? Some companies say, oh no, we'll test it, and when we can find nothing in it, we'll dump it to the natural, the local, whatever they call it, environment. In a lot of the cases, you actually can't detect it. So, you know, that's because it's new, yes, and because the EPA is not on top of this at the moment, yeah, that's a risk that's out there. So I'm just sharing information, okay? So ADC facility, look, you probably won't be able to read this, but what I was trying to show in this slide was when you're designing one of these facilities, yeah, there's lots of factors. There's sustainability, but the challenge with sustainability, you need to, what did I say here? You do need excessive use of water and chemicals, you know, to clean, yeah, to make stuff safe. For example, if it's a filling line and you're filling this highly potent stuff, you can't just take parts of the filling line out and put it in a parts washer. You need to rinse and rinse and rinse inside that isolator to get some level of assurance that there's no free powder or anything like that on the line. Then you bag it out, you bring it to your parts washer, yeah? Somehow load that parts washer safely without releasing anything into the air, without the operator wearing breathing air, by the way, because if they do, you've failed, okay, because that's your first line of defense. And then you have to wash and wash and wash and wash if you need to reuse that equipment again. So what we find in these ADC facilities, we try not to reuse equipment. We try to focus on single use. Get rid of it, incinerate it, okay? So these slides will be made available, and I know people want to go home. All right, so if we go to this, so this, what I'm trying to show here is there's MABS manufacturing, okay, involved, there's payload linker and drug linker synthesis going on, there's conjugation, and there's aseptic filling and freeze drying, external vial washing. You have to do external vial washing because it's highly potent, okay? Inspection and secondary packaging. They're all the unit operations. Now, there's loads of guides out there. So if you're looking at, so I use the ISP guides, they're great. Okay, so there's the ISP guide for API. Okay, so that'll deal with the drug linker. There's the biomanufacturing facilities, that'll deal with the conjugation. And the sterile product manufacturing, that'll do with the fill finish bit of it. And then if you're talking about containment, there's tons of containment guides out there as well that you need to read. And these are quite difficult to read, some of these documents. So if you have a 1 nanogram, or say 10 nanogram OEL level, you need to, when you're buying a bit of equipment, the vendors will need to demonstrate to you that their equipment will meet those requirements, and you typically test to 10% of that they'll be doing tests to 1 nanogram containment. There's very few vendors out there that can do that. So you have to do a lot of due diligence. So I think yesterday somebody was talking about isolators for high potent containment. There is a company actually in the UK, Solar Containment, which is part of ILC Dover, the American company, yeah, and they do flexible isolators for high containment applications down to 10 nanogram. And we're buying a lot of them for high potent applications because you don't have to worry about cleaning them or anything. Solar Containment are experts in high containment applications, as are ILC Dover. And that, having one of those where you use it, you vacuum it up, and you send it off for incineration versus having a very complicated isolator where you really can't demonstrate it is free of the potent material because you can't really test it to find traces of that high potent. I think it's no-brainer, but we do have certain clients, I can't mention the names, they are American, they are top-tier pharma, and they won't accept flexible isolators for high potent. Now, when you're going down to 1 nanogram, like Lonza are doing at the moment, plastic won't work for that at the moment. Okay. So what I'm going to talk about quickly now is some of the designs, just to give you a flavor of what's out there, what we've done, what we've been involved in. Yeah. And look, the thing is, primary containment is typically the item of equipment. Secondary containment is typically the room. So it could be the reactor as the primary containment. If it's a freeze dryer, it'll be the freeze dryer plus the isolator that's around it because you open the freeze dryer when you have when you're trying to unload it and load it. Disposable versus reusable equipment. I'd always propose reusable equipment unless it's not suitable for the application. Some conjugation reactions and the solvents involved and stuff like that, they're not suitable for reusable. Cleaning, decontamination, deactivation, your approach to that. Are you going to use stuff once and just get it out, okay? Are you going to try to decontaminate or deactivate? You can deactivate some of them, but very few of them, yeah. And there's vendor experience and expertise. That's key. You really have to spend money on this. And then there's multi-product flexibility. A lot of these facilities we're designing, in fact, all of them, none of them are single product. So the clients are saying we want to build into the design multiple flexibility, but we actually don't know the process. Yeah? So we just have to make the rooms big enough and make sure the unit operations will fit in the rooms. Yeah? And the scale of operations. I mean, the scales, look, it's all small scale. It goes from 10 litres up to maybe a metre cube. So they're small scale. Regulatory compliance is a big thing, which I talked about, the different regulatory compliance requirements for the different phases of the unit operations. And there's new drug conjugate modalities. Because I talked about antibody drug conjugates. There's peptide drug conjugates. In fact, we're doing a job in the States at the moment. It's $1 billion job where they have antibody drug conjugates and peptide drug conjugates in the same campus. Right. So this is one job in the US. So I'll just show you, this is the layout, okay, first. So what we have, this is kind of a ballroom, yeah? So most of the unit operations of conjugation and MABs. Sometimes with the MABs you need to modify it slightly, yes, before you introduce it to the conjugation. That doesn't happen for this process here. So the MABs will be brought in, it will be thawed in the room, then you'll have your conjugation vessel, you'll have your drug linker, which will be dissolved in some solvent and then pumped into your conjugation vessel. Then you'll throw in your MABs. Then you'll add some chemicals. The conjugation will start off. It's kind of a black box what's happening in the conjugation vessel at the time, but it's all happening in this room here. Yeah. Once it's conjugated, then you have a bit of purification. You can have TFF, you can have chromatography, you can have both. Yeah. And sometimes you have dual payloads as well. So it's not just one payload, you'll have two different types of payloads. So you have two conjugation reactions going on. Yeah. All happens in here, and then you do a bulk fill into some kind of bottle. Yeah. Here it's in the C room. Sometimes people want it in a B room, into an A container. It's nonsense, but that's what some people do. All right. And what you'll see here is that the big thing, this is the containment boundary. Now, Justin in a few minutes is going to talk about airlocks and getting into these rooms and getting out of these rooms and misting showers and all that stuff. Yeah. So what I was trying to say here, it's 10 nanograms, it's multi-product. They don't know the products, actually, okay? It's disposable technology. Now, they wanted to allow for future rigid isolation technology because they weren't sure whether the flexible would work, you know, if they went lower and stuff like that. And they've no clean out of place, they've no parts washer, they've no autoclave because they're going to chuck everything out once it's used. Yeah. So the rule of thumb when you're dealing with high potent stuff, if it doesn't need to be in the room, don't bring it into the room. If you're making up buffers, you don't bring the buffers in the room, you pipe them through the room, and you make sure when you're pressurizing the room, you don't pressurize from the high potent zone to the safe zone. It was the basics, yeah, okay. So this is another layer here for a job in America, and what we have here, that's the MAB suite. So it's a dedicated MAB suite because they're doing stuff to the MABs to get it ready for conjugation. So there's some chromatography and purification of the MAB in that room there. That's their buffer prep room, so that's all outside the containment zone. Then they have the conjugation suite, and then they have their bulk fill suite there, and they have a bit of weigh and dispense up there. So again, that's a different layout, yeah? It's conjugation, but it's a different layout, okay? See if I've, so we have, what's this? This is one in Europe, okay? Try what this was. In fact, this is, a ballroom design. I want to get you a layout here to look at. Yeah, this is one very close to home, okay, in the UK, all right? And this is a buffer prep zone here, and this is a conjugation room here, and everything is happening, you know, in the conjugation room. This is a contract manufacturer here, and it's 10 nanograms, all right? So now this is an API synthesis plant that we're doing in Europe, okay? And it was an existing shell space that we needed to stick a whole load of equipment in. So we have glass reactors with lots of solvents, nasty solvents. We have flexible isolators. We've done it over 2 floors. So we have reactors, we have freeze dryers, we have tray dryers, we have chromatography, and we're all trying to squeeze it into a very small area, and the client has very little money as well, and the space is very compact. So it's not ideal, but this is, so we had lots of challenges in this one, yeah? But it makes it interesting because there's no Annex 1 applies to this and stuff like that. All we need to do is to keep containment within the zone. Okay. Now this is a fill finish facility here with conjugation. So what you see here, this is fill finish. This is a formulation, and over here is a conjugation suite. Okay, and it just happens so there's 2 facilities in one, and that's 10 nanograms, down to 10 nanograms. So these are the considerations for these type of facilities: flexible versus rigid, active containment isolators, extract booths. Some people, believe it or not, stick their unit operations like chromatography and UFDF all within extract booths. Some have them sitting in the middle of the room. So, I mean, it's the same containment requirement, but they're all different. And it's just the way it is in the industry at the moment. Breathing air, some have it as standard, some have it by exception. If you're dealing with solvents, you need to do ATEX risk assessments, which is a bit of a challenge. So you got GMP, you got high potent containment, and you got ATEX all in the same suite. There's multi-product and multi-modalities that offers a challenge as well. And some people want potent and non-potent in the same suite. You can do it, but obviously you just need to risk assess how you do it. Yeah. And there's sterile and versus bioburden control, as I talked about in the bulk fill, and the room grades as well. There's no consistency in room grades. Right. So now we get to the interesting bit, Justin, architect.
Thanks, Kevin. Okay, I will run you very quickly through the clean room operating as an engineering control for this. And I think Kevin touched on it very well there. You know, when we do this work, we have to cooperate really tightly with the process engineering and the process technology group. That's why there's two of us here. We can't treat each other with the distance we used to. We really have to work really closely together. And it's because you keep getting the consultant's answer to everything in these. It depends. It depends on the risk assessment. It depends on the equipment. It depends on the operations. But I think Kevin covered it again accurately. Engineering controls is what we're interested in here. For various reasons, we can't do the top and we don't want to do the bottom. So this is a diagram that everyone will be very familiar with from normal GMP operations and from room classification. You know, you've got your critical grade AB area here, and you go out through your C, your D, and out to outside. We've got the same thing. This is out of the ISPE guide, where we are relying on multiple layers of containment. I'm going to go quickly through this, but your primary containment is your closed process, whether that's a vessel or whether that's an isolator, depending on where you are in the process that was described there, from the API through the conjugation down into the fill finish. There will be different primary containment, but there will be primary containment. Secondary containment is something that's been around API forever. It's been around potent substances forever, but it's much, much more critical now because of two things. One is the very high levels of potency that we're using, and the other is it's not always detectable. We can't assume that there hasn't been a breach within the primary containment. So your secondary containment really does become a massive part of how we design these facilities and how we think about them. Tertiary containment, you know, you don't want to build this thing in the middle of a very sensitive area. So what does that mean? Again, this is the ISP guide. I hate the colors because they're the same as, they're the opposite to what we use for GMP colors. But green is safe, yellow is hazard, and red is danger. So when you go through that, what does it actually mean? It means the red zone is contaminated. You expect the red zone to be exposed to potent compounds. It's dangerous. You cannot have humans anywhere near that. Yellow is your secondary containment area. So this is, it shouldn't be contaminated, but it's going to get contaminated if there's a breach. And if there is anything out of the ordinary happening, that's what you're relying on as your secondary. And then when you get out to this manufacturing corridor, we tend to forget about it, but it is worth thinking about. You can have an open corridor there with people in the office and wandering out to get their lunch and to buy a sandwich. It's not necessarily whatever type of facility you're in. This may not be behind a garbing area. So, you know, you're not very far away from danger, and it is important to get it right. So how do we do that? We do that with airlocks, which is what we've always done. Airlocks give us a little bit of a contradiction. There's always a contradiction between containment and GMP. But generally, you're changing garb to go across a classification boundary, you're changing garb to go across a containment boundary, or you've got an improvement step if it's materials. So the airlocks tend to become dual purpose. We're trying to get an airlock to do two radically different things. So on the left, we have our traditional GMP. You've got your production area up the top. you blow the air out towards a progressively dirtier area, and everybody's happy. That works for everybody, and all our risk assessments and everything say we're doing the right thing. But if we have a potential contaminant and we're blowing it out into the safe corridor, that's not going to work for anybody. So the obvious solution is you turn it around and you make a sink. Blow all the air from the corridor in there, and that protects the corridor, and you've still got your airflow out of production. And that works in some scenarios. The difficulty with it is our friend Annex 1 tells us the airlock has to operate at the same standard as the production facility behind it. Doors are very seldom proven to be 100% sealed, and anyway, when you open the door, you're dumping a load of lower grade air into that airlock. If you've got particulate monitoring or anything in there, the alarms are going to go off. But there are times when it is appropriate. So if you try and solve that again, you go to your bubble here, and you can blow the air back into production because we can now classify this airlock as the higher grade because it's flowing out into the corridor. And this, again, has some scenarios where it works. Where this falls down comes to the operations in the airlock. If this is a mal out and you're taking material out of your potent zone and you're doing any sort of operation that could shed anything in that airlock, you're going to blow it out under the door. Or as soon as the door opens, you're going to whoosh it out into the corridor. Again, not what you want to be doing. So if you've got the money, if you've got the area, you build a bubble and a sink, which is a great solution. Your sink is giving you that break for your contamination. So all contamination comes in there. If you've got air wash showers or misting showers or anything, they're generally in the sink. You can now put your containment boundary on the end of the sink, and now you've got a GMP bubble at the end that's protecting the corridor and it's protecting your production zone. So if we expand that out a little bit, again, ISPE guide, this is an idealized airlock strategy. This is what we'd do if we'd all the space and we'd all the money all the time. But it is a very good illustration of what can work here. So we've just drawn on top of this to illustrate what it can do and what it can't do. To think about this in any sort of rational way, we've got to consider the containment boundary. So this is a containment boundary superimposed on it. The easiest possible one is your mal in. When you're coming in through the mal, there's absolutely no reason to believe that you've been contaminated in any way. So the containment boundary sits on the inside. When you're leaving the facility, there's every reason to believe that you have some contamination, so you can't cross that containment boundary until such time as you have changed your garb or gone through some process of, yeah, I'm talking about garb and mals, but you know what I mean. PAL in, exact same discussion as the mal in. There is no reason to believe that you've been in any way contaminated, so you don't need containment. You're not crossing the boundary till you get there. Again, this is all secondary containment boundary. When you're leaving as a person, you're going through what's called up as a fogging or misting shower here, but that's your D-garb. That's your sink. That's where you're getting rid of anything potentially contaminated off you. You can then cross over and be out into showers and gowning and whatever else. But we then start to think about this in terms of our HVAC, because it's absolutely critical, and it becomes that discussion or that description I was giving you earlier about how these things operate. So this is just a very quick illustration. You know, the bubble, we were saying, there are issues with it from a containment perspective, but where the containment boundary is over that side, use the bubble. There's no need to have a sink in there. It's quite an effective control all by itself. The mal out in this one is shown as being a sink only. How are we getting away with that? Because it's out only. That airlock doesn't need to achieve the classification. Nobody's ever coming back in. So in that instance, you can run it as a sink. Do the easy one. Coming in through the PAL in is the exact same as the mal in. You're not crossing the boundary. You can operate it as a bubble. Then, when you're going out, you're hitting that sink. You get rid of all your garb in there, and now you're going against the airflow, and you're out. So when you open that door, there's nothing coming in, there's nothing coming out. Everything is coming into the sink, And once you get out here, you haven't brought any contamination with you. And then you're clear. That's set up as a bubble there, you know, where you've got additional rooms there. Unless you have a GMP reason, they don't necessarily need to be considered as part of the containment strategy. So, and I'll finish with this, and we said we'd do it on a bit of the lessons learned bit. The consideration of the operations. Everything I've said there is very generalized, very non-specific. When you're doing this in facilities, you've got to work really, really closely with our process and technology colleagues, with the client quality teams and the client EHS teams, because every client will have a different approach to this, and every client will have a different product, which will have a different exposure danger level. And it depends what you're doing. You know, when I see manufacturing suite, I always think of fill finish, because that's what I started doing. But that could equally be the API area. That could equally be the conjugation suite. And they're very different activities in those with different levels of risk and different approaches. So that's the one critical thing. The other thing we just said we'd mention it is, because you're at, because this is Cleanroom, and we're at such low levels, this will occasionally be run at negative pressure. I could talk for half an hour on that, I'm not going to, but just be aware when you're building clean rooms and running them at negative pressure, or when you have positive pressure with potential contamination behind it, it needs to be gas tight, and it needs to be tested to be gas tight. So that gives us a whole set of other headaches. Mastic is not an appropriate material generally. It will only last a couple of years. It's not the right thing to be using. Where you've got penetrations, you need clamps, you need gas-tight connections, all that through it. I could go on at length, but I won't. I'll give it back to Kevin to talk a little bit about the markets and what's happening. That's us, I think.
Yeah, thanks for your patience. I'm going to be really quick. I'm going to speak really quickly. But for containment, so Justin did talk about airlocks. It's a bit of a head wrecker because they're all different and the approaches are different. Bubble sinks, you know, whether it's a bubble and a sink or whether it's a cascade. There's no right answer. You just need to follow the potent. But containment boundaries is a key thing. So when I'm buying sterile equipment like freeze dryers or filling lines and stuff like that, what I try to make sure happens on the drawings, the vendor drawings, is you identify the sterile boundaries, especially for freeze dryers. When you're buying an item of equipment like a Lya that's dealing with high potent stuff, not only do you have to draw the sterile boundaries, you also need to draw the containment boundaries on the equipment, and the vendor has to demonstrate how they ensure potent material from your vacuum pumps, for example, in your freeze dryer, where they stop that. Do they have a filter downstream on that, and how do you maintain that filter? And maintenance is a critical thing. When you're doing clean rooms on your secondary boundary, it's all full extract, okay? No one's brave enough to recirculate it back into the room yet, but they might do with all the sustainability stuff going on, right? But it's full extract, but then your low-level extracts, they need to be ideally within the room. You don't want to contaminate the ducting, but believe it or not, some of our clients don't want low-level extracts in the room. They'll stick a HEPA filter somewhere in the ducting. That's just real world. That's what's happening out there, okay? So the market update, what I recommend you all do is, Fierce Pharma is a great website, okay? It gives you an update of what's happening in the pharmaceutical industry. And this is just from Fierce Pharma in January. I think it was January, all these things, okay? Seagen, so I mentioned Pfizer had spent 42 billion on Seagen, okay? But Pfizer are still spending more money, okay? Merck, that's the American Merck, have spent 20 billion, okay, on a deal with Daiichi for ADC conjugates. J&J is beefing up its acquiring Ambrx for 2 billion. We have now, Novartis are not going into ADCs, they're going into radioligand drug conjugates, okay? So instead of a HPI, they have a radioactive bit that gets into the cell and does the work, okay? And then this is AstraZeneca, this is 2 days ago, okay? That 1.5 billion manufacturing facility in Singapore. So hopefully IPS will be doing that job. Singapore is a great place to work, okay? So the trends and challenges and stuff like that, just as a quick summary. The campus concept. I had a presentation to a client there a few weeks ago in the States, and they said, why don't we do everything on the same campus, Kevin? And I said, that's a great idea. And you know why it's a great idea, in my opinion? Because there's very few people out there who have experience and expertise in handling high potents in a GMP environment. So if you can control everything, it's a good idea. And lo and behold, AstraZeneca took my idea on board and investing $1.5 billion, okay? And there's new technologies out there for ADC. There's continuous chromatography and there's spray drying. So things are changing all the time. And look, there's waste generation treatment. I talked about that. There's a lot of mergers and acquisitions going on. And flexible facility design, which is always a challenge. And there's other types of drug conjugates. There's pretty pictures of some of them, and there's pretty pictures of other types. So there's a whole load of drug conjugates out there. So, Sinead, thank you.
Kevin, Justin, thank you very much. You've described a very complicated facility. That's really good. Thank you very much for that. We've got any questions for the guys?
Yeah, just
Yep.
one more you answered right at the end, which was on the negative pressure, which I was kind of expecting it to be. But I'm interested how you get the waste out of the PAL sink and the garments that they've degowned at that point. There's a waste management out of this facility as well, or garment management out of the facility. Can you maybe explain it from the layout diagram?
Yep, we can try. I'll go back to that idealized one, if that's all right. So you have two things there. One is where you are degarbing. So that is generally done in a misting shower. Again, I think it's come up in the last 3 presentations. The industry does stuff that isn't always very well thought through. So we have mist showers on the exit through containment boundaries. A mist shower for a liquid product does absolutely nothing for you, apart from get you wet. But I think every one of these we've done, we've been told to put a mist shower in it. One of the things a mist shower does is a continuous bag liner out. So everything that comes off gets bagged immediately and is sealed and can be heat sealed. And it generally comes out that corner there. Yeah, it's...
In my brain, what you're going to have to do, but how do you get it clean? You can't detect. You can only sterile wipe the back, can't you?
Yeah, but it's a continuous liner. So the inside of the liner is open to the containment boundary. The outside has never been in contact with the containment boundary. And then you can tie that or heat seal it, and then you're confident. You generally double bag it immediately, just so you don't catch it in the door handle going out or anything.
The challenge is detection. And to be honest, when I hear what's happening in the real world out there, it's amazing.
Shocking.
All we can do is make it better. Okay.
Thank you. Any more questions? Going once.
Very much.
Going twice. Okay. Thank you very much.
Thank you.
Excellent. Thank you very much.